Okay, there are four structures to a protein. We have the primary structure, we have the secondary structure, and then we have tertiary, and then we have quaternary structures. So the first structure is the primary structure, and that's defined as it's the linear sequence of amino acids. So it's like the arrangement of the acid sequence in one single chain. So the bond type is it's made up of covalent bonds, and we're going to call those covalent bonds a peptide bond. So covalent bonds are very strong bonds, and the peptide bond, essentially, it's each amino acid together in a sequence is formed by those covalent peptide bonds. And a whole chain of them connected is called a polypeptide thing, which is what proteins are. So essentially it looks like we have an amino acid connected with another amino acid. Okay, and then these bonds that are formed there, that is your peptide bond. So between each amino acid, there's a peptide bond connecting them, and they are covalently bonded together, which is a pretty strong bond. So the parts of amino acids that are involved, well, the first thing we have to talk about is what the amino acid looks like. So amino acids they basically look like this. They all have one singular hydrogen connected with a carboxyl group, which carboxyl groups, they look like this. You have the double bonded oxygen with the and then so each amino acid always has, this is the carboxyl group. It has your amino acid group. It has the one singular hydrogen, and then it has the R group. The R group can be basically it's anything. There's about 20 of them, and the R group is essentially what makes the amino acid its characteristics. It characterizes the amino acids. So this could be different amongst any of them. A peptide bond is formed when an amino acid and a carboxyl group join together, and this is called a dehydration reaction or a condensation reaction because you're losing H2O. So I'll draw what that looks like for you. So it's taking the hydrogen from the one amino acid and then the oxygen and hydrogen from the other one, and these will go out H2O, and then this is the formed new thing, and it's going to be called a dipeptide because it's two of them combined. So it looks like okay, and so that's called the dipeptide, and that's the new form, and that's formed again through the dehydration reaction because, again, we took out the water. So this bond, the strength of it, again, this is a covalent bond. Covalent bonds are very strong, so the strength of the bond is very good. And then the overall strength, it's very, very hard to break down the primary sequence of a polypeptide because of these bonds. Okay, so the second structure is the secondary structure, and that is defined as the way the linear sequence folds upon itself. And this is determined by backbone interactions, which is also determined by hydrogen bonds. So, two examples of secondary structure is the alpha helix and the beta pleated sheet. So, they look like this. This is the alpha helix. And then we have the beta pleated sheet. And it looks kind of, well, this is how. So the alpha helix, it has like more of a coiled shape to it. So it looks like this, in a sense. And then beta sheets look more almost kind of, so this is like a coil, and this is one's almost like an accordion, if you can imagine it folding in on itself. And so these two are, it's carbon nitrogen, the backbone to it, and the R groups are like attached on the outside. It's the R groups are not involved, it's the carbon nitrogen backbones interacting with each other. And the type of bonds that happen here are hydrogen bonds. So it's the carbon nitrogen backbone, the R groups sticking out to create these hydrogen bonds. So for the A helix, the hydrogen bonds are going more up and down. They're kind of connecting this way with each other. And then with the beta pleated sheet, they're connecting like this way with each other. And that's like move it around. Okay, so hydrogen bonds are relatively weaker bonds. So the bonds between like just a single hydrogen bond, it's fairly a weak bond. But the overall strength of the secondary structure is actually very strong. It's weaker than primary structure, but it's actually very strong because remember, when you have lots of hydrogen bonds together form a very, very strong bond. So overall, this is a pretty strong structure. So the third one we're going to be talking about today is the tertiary structure, and that's defined as it refers to a higher order of folding within a polypeptide chain, which is essentially saying it's the final 3D structure of a single polypeptide. So the bonds that are with it are, we have hydrogen bonds, we have it's disulfide bonds, we have ionic bonds. It's called van der Waals attraction. And then we also have the hydrophobic bonds. Okay, so the difference between tertiary structure and secondary structure is that secondary structure involves more of the carbon-nitrogen backbone connections between there. Tertiary is all about the R groups in this one, so that's the parts of the amino acid that's involved. It's the R groups. So with these bond types, we have the hydrogen bonds, we have disulfide bonds, which are actually covalently bonded. Their bridges can form between two cysteine side chains. Ionic bonds, they form between groups of opposite charge, and then the van der Waals are weak attractions between atoms due to oppositely polarized electron clouds. And then the hydrophobic bonds, it's essentially saying you have like, it's in an aqueous environment, so it's like water, we can just say H2O's around here. So all the polar groups of it are going to come to the outside because they like the water, and then all non-polar are going to be on the inside part because they don't like the water. So that's what the hydrophobic bonds is going to be like for this. And the strength of all these bonds, they're mainly weak. they're fairly weak bonds except for the disulfide bond right here. This one is a little bit stronger than the other ones because it is covalently bonded, and covalently bonds are stronger. But overall, the strength of the tertiary structure, it's overall weak because these other bonds are fairly weak as well. Okay, so for the last one we'll be talking about today is the quaternary structure, and that is defined as bonding between multiple polypeptides. So essentially it looks like this. So you have like one polypeptide, it might have like the alpha in it, or like the beta pleated in it. This is the one polypeptide, but quaternary structure is talking about how multiple polypeptides interact with each other. So if we have like, we can have 4, we can have 6. It's just multiple polypeptides, more than 2. So essentially it's saying it's the final 3D shape of 2 or more polypeptides. So the bonds that are associated with this, it's the same as the tertiary, and the parts of the amino acid involved are also the R groups, same as tertiary. And so the overall strength of the bonds is still relatively weak, and the overall strength of the quaternary structure is also overall weak because of those same bonds as the tertiary ones. So essentially this is the final structure of the protein.